There are many different species of corals. They can be found in different colors, shapes, and sizes. Although being made up by tiny marine organisms called polyps, they can create colonies that can be seen from space. But not all species of corals build coral reefs. Those that do are called hermatypic corals, belonging to the phylum Cnidarians, like jellyfish or sea anemones. They generally exist in a holobiont, a complex microscopic network with bacteria, fungi, virus, or dinoflagellates, sequestering up to 90 million tons of carbon per year. So, how much is there going on that is missed by the eye? The majority of biological activity happens within a thin layer of coral tissue. Reef-building corals live in a symbiotic relationship with single-cell dinoflagellates called zooxanthellae. Zooxanthellae gives up to 95% of energy to the corals, being essential in the construction of the skeleton. However, when the temperature of the water rises, the excess of energy is turned into reactive oxygen molecules that damages the cells of the polyp. Then, the polyps expel the zooxanthellae and the corals bleach. The increase in the number of disease outbreaks and mass bleaching events due to rising sea temperature and ocean acidification have massively declined the population of corals. To understand the micro-scale interactions between corals and their environment, this paper presents a new method that they call coral on a ship.
In this experiment, the researchers attempted to answer the three main problems which are faced by coral reefs. These are acidification, bleaching, and pathogens. Before they explore this, the corals are micropropagated into the slips through polyp bailout. This whole process will take over a week. The microslips are then set up with one-way water flow to control the conditions and to let the coral acclimate and settle. They observe the polyp and subject it to time-lapse image using DIC every 10 minutes. They are on the lookout for early signs in development of spinules and septa. These are a key stage of calcification. If these are present, the polyps are healthy and microscopic representative of the mother colony. Observing calcium carbonate crystals requires staining with the use of calcium dye. The dye incorporates into the lattice by using SEM. They were able to image the coral and observe the intermittent heterogeneous growth. These green dyes become very concentrated where growth rate is high and calcification is undertaken. After a week of healthy growth, the conditions are gradually switched to semi-lethal to induce coral bleaching. This is done by simply increasing the light intensity to eject the symbiont out of the cells. As a side note, this emphasizes the importance of using low-intensity visible light methods such as DIC and using the natural fluorescence of corals and symbiont to an advantage. Because of micro-on-a-chip method, they were able to look at the symbionts up close and even into the single-cell levels. Interestingly, symbionts ejection is independent of proximity to each other. The researchers concluded that different bleaching kinetic levels were results of localization within the polyp and variability in ROS. Furthermore, they noticed that before ejection of the symbiont, fluorescence is at its highest until it disappears. Lastly, an coral pathogen, Vibrio coralliticus, was transformed with a plasmid encoding for red fluorescent protein. This made it easier to observe the pathogens and the pathways they take and where the corals are most vulnerable. There are many positives with using coral on a chip method, and combined with modern technological advancements in genomics and satellite imagery, we may be able to use this to protect our corals through modifications or by careful monitoring from outer space. Of course, there are also other applications other than corals. Important commercial farming where a miniaturized controlled environment is a great tool before the large scale and expensive trials. Thank you for listening. We hope you enjoyed.